Our next speaker is Blake Fitch with Max Planck Institute for Biological Cybernetics. And his talk is MrData, an iRODS-based human research data management system.
Thanks. I'm Blake Fitch from Max Planck. It's the Max Planck Biological Cybernetics. Basically, we're a neuroscience institute, but we're not a simulation neuroscience institute. We're a human research and wetware type institute. So there's fish and mice and humans. So where's page? This is like the other kind of keyboard.
Yeah.
Okay. So we're, in contrast to the others, we've been doing iRODS for a little over a year now, and this is going to be a kind of a talk about where we've gotten to. Our first target is medical imaging systems, in particular MRI systems. We have a Siemens 9.4 Tesla machine, which is one of, I guess, about 3 in the world. It's not a clinical setting, it's strictly a research setting. The components I'll draw your attention to are Castellum, and then the MrData system. I don't know if I have a pointer. Can I mouse on this? Okay, and the MrData system, which I'll get to, is a set of processes running in containers. And we have, of course, scientific investigators, and our backing store is GPFS. In terms of access options, it's the usual MetaLinux for browsing, Python, iRODS client in Python. We found our clone to work. We tend to have fairly large files or fairly numerous files in data sets. And we tried to use CyberDuck, and they had some caching issues. So that we're pretty solidly in our clone through the WebDAV interface. One of the things that we took very seriously on this project was the separation between the data archival system and let's say the human subject metadata management, address, phone number, contact information, details about whether the person lay still in the MRI machine or fell asleep. They're not supposed to do that. That all has to stay in a secure and private system. And so unlike some of the prior work in medical image archival, we wanted to use completely separate systems. And this Castellum system came up very early in our development, and they this is a group out of Max Planck Berlin, and for anyone that needs this kind of human subject recruiting, safe, GDPR compliant, I've got the link there. You should check them out. They're open source. They'd love to have more people using their stuff. But basically what you do is inside this system, you define studies, and then you recruit subjects for the study. And all the private information stays in this system, and only pseudonyms, so alphanumeric tags for this. That's not what I meant. There's an argument for doing it off your own machine.
In this slide?
No, way back.
Oh, way
Back.
Yeah.
Can I arrow down?
Yeah, the arrow.
Excellent. One of the things we worked out with them is an application programming interface so that we can extract approved data for export, attributes about the subjects that are not private, but are needed for scientific data management. And this brought up something that was a little challenging for both of our groups, which is that there's this mixed use metadata. The Castellum people were very focused on making this recruiting metadata safe and putting out pseudonyms. And we were very focused on the data archive and automating the uptake of the image data from the MRI scanners. But we also needed things, metadata in the scientific archive that's necessary for recruiting and therefore must be homed in Castellum. And that's for MRI research, that's handedness, the languages that you know, how good your vision is, roughly your age, although not exactly, but name and address, date of birth, this investigator shouldn't know, and the data management system shouldn't know. And then there's a bunch of other scientific metadata that, as you'll see, we pick up out of the header files. So what pulls all this together is a single web page that we have left that involves putting in an experiment owner. So an experiment is a single session on the MRI machine, and with a single subject and a operator or investigator. And but we don't want to put the person's name in at the MRI machine, because then all the medical imaging data files will be corrupted with their name. So in a clinical context, that doesn't matter, they put your name in. But in our context, we don't want that in the data files. So you enter your user ID, you enter the subject pseudonym that you got from Castellum, you put in a study ID, you push submit, and you get this alphanumeric here, which you then use to, when you go to the MRI machine, you type that into the patient name field. So this is the basic workflow of a MRI research investigator. Use Castellum to define a study and get a study ID. So just going back, that's the 22 here. And a study is a project, roughly speaking, for, to map the other nomenclature I've seen. Use the GUI to recruit subjects to participate in the study. That's searching, emailing, phoning, getting responses, whatever it takes to line up the human subjects. And then acquire a pseudonym for each subject in the study. And then you go and register the experiment on that web page that I just did. You get a study ID, you put in a study ID, and you put in a pseudonym, and you get an experiment ID. And when you do that, that metadata is reflected into iRODS, and we also record it in a GitLab repo. At the MRI scanner, so here, the operator has a console where you could put in a lot of personally identifying information, but again, you only put in the experiment ID, and you run the scanner and DICOMs, which are typical medical imaging data, and Twix raw data, which is a Siemens proprietary format, come flowing out and are processed by an importer or uploader function, and the DICOMs and raw data go into iRODS. So our implementation, this is a process diagram of the same stuff. So starting from here, we have an MRI console, and you put the experiment ID in there, and the scanner runs, and we have a landing zone in GPFS. Unfortunately, the scanner requires a SMB or Samba landing zone, and these processes here see the same region via NFS. And they, for each data type, the DICOMs, the TWICs, and extra data, which is a directory that the scientist can populate with whatever they choose. Each one of these uploaders for the context gets metadata from iRODS so that it knows the study and it knows the study ID in particular, and catalogs and extracts metadata from the headers. So how is the machine configured in detail, which will be searched for later, and commits those DICOMs to iRODS in that way. And here we read the header of the raw data file and commit that to iRODS. And here, this is just a free-for-all directory that the scientists can populate with whatever stimulus data or other logs, whatever they like. And we mirror that directory structure into iRODS. Here's the forms process, which puts up that one web page now. Used to have 2 or 3. Here's the Castellum to iRODS, and this is polling the Castellum virtual machine, which is outside of the kind of MrData host boundary. And this is an investigator workstation where they might use rclone or other browsing techniques, usually targeting DAVRODs in our environment. Here, providing the pseudonym and study ID, getting back the experiment ID, which can come over here and drop into the console. And this is a polling interface so that we poll Castellum for all the active studies and represent that in both iRODS, and we keep that metadata historically kept in a private GitLab repo. So this is everything that we've done in the MrData system itself has been implemented as Docker, a set of Docker containers that run on a single host. At our site, if the host has Docker, we don't allow any end users on that host directly. We consider it, you know, sort of administrative context. These Docker containers are deployed with Ansible's Docker module. We had done a Docker Compose in implementation of this, but Docker Compose has a trouble where it sort of assumes you're on the Docker machine. I mean, there's some remote things you can do, but when you use Ansible, you can actually poke at the host machine and set up file systems and make connections, and then bring up, say, iRODS under the control of Ansible, and that's what we do. And this is one of the core pieces that we're thinking about open sourcing, because I think it would be nice to have, having fought the battle of getting iRODS into a container that I use in production, it might be nice to share that if other people are interested. And we haven't got full CI/CD continuous integration, but we've designed toward that end. Already what works is, so what you're looking at here is our bare metal Rocky 8 production host, but the way I test is I have a Rocky 8 virtual machine, which I have a little script that clones and wipes it out, and then you just run the Ansible into it, and you have the entire whole system reproduced for testing, and that should be amenable to continuous integration. And the other good thing is that many of the iRODS ecosystem processes, MetalNX, WebDev, they come kind of with an almost ready to use or sometimes ready to use Docker implementation. So you can start picking and choosing and pulling pieces in that way. And that's been very nice. I could comment that the reason the first implementation, every one of these was in its own VM. And our problem with that was VM IO bandwidth. And this is the rough layout in our iRODS vault. You'll see a lot of it looks boilerplate, but here's the study ID. And here is the experiment ID. And so there are 2 experiments here. And then each data type for one of those uploaders has its own directory. And then the contents of those directories or collections are dependent on the type. And we are placing unified, well, AVUs that are representative of all the TWICs in this session, in this experiment, would go on to these collections here. We're trying to kind of propagate up where the AVUs are so that we don't replicate them on small or numerous files. I think that's it.
All right.
Almost got you back on schedule.
Right. Thanks. Any questions for Blake? Yeah, can we get a mic up here?
You only need 2 mics.
So you showed this Python import function that you use. So what happens when the output data from Siemens changes? How do you deal with it?
Well, DICOMs are very unlikely to change. That's a long-term standard format. It's a horrible format, but it's long-term and standard.
Right.
So reading, you can find Python to read those headers and that kind of thing. It's unlikely to change. The TWIC's raw data is like, it's raw K-space data that, you know, it's the other side of an FFT from an image. Siemens can change that at will. It's like, my paper can't find a reference for it, but if you Google it, there's loads of people that know about it and process it. We just have to, I mean, it's still a research environment. We would have to suck up any changes in that. But that said, there are many research groups that are getting into those files, so their changes probably won't go too fast.
Right. Thanks.
All right. Any other questions? We got another down here.
How do you automate the data data flow from MRI to the MrData? Do you use the facilities of iRODS like auto ingest tool or something else?
It's a good question, and we don't. We might perhaps should. It's a really, good question. The history of this project is that a lot of that started using a MRI image management system called XNAT, and we looked at another one called LORIS, and we started finding ourselves implementing a lot of scripting around those other solutions. And then when Castellum came in, it kind of overlapped with those solutions so much that we moved into the current configuration. I should investigate that, although I will say that there's a lot in there, including heuristics about the timing. So DICOMs come out of the scanner automatically into the landing zone, but they don't tell you for a given scan how many DICOMs are going to arrive. So I have logic in there that says, okay, if the next scan starts, bundle up those DICOMs, right, and ship them. But it can be that another late DICOM arrives because there's no part of that that's it's not a kind of complete signal, right? And so there's heuristics in all of these things that might be complicated to implement, and this is the implementation I have right now, but I am open to looking at automating more of it for sure.
The second part is the metadata extraction from the DICOM files. So this is, I guess, also not done in iRODS. I mean, when you upload data in iRODS.
I think I've seen something out there when I've, been researching this, that people have done that. But again, I was pretty far down my own path with the Python library for DICOM. And I mean, this is a one-man half-time project because I take care of the HPC cluster too. So there could be more automation native to iRODS that does DICOMs. On Twix, I rather doubt that because most medical clinical contexts never look in Twix, right? You'll never get one if you go have an MRI. And the header, the Python that reads the header is just some researcher who keeps it bubbling along and updated. The extra data directory, the expanded data directory, I mean, the only heuristic is when is it done? And so we have kind of two ways of dealing with that. After 24 hours, you go pick it up. Or the person can drop a finished file in there and then the uploader would pick it all up and catalog it. And probably that can be done with other tools as well.
Yeah, we had a question over here.
Hi there. I noticed you encoded the study ID and similar in the collection path. So can you let me know why you decided to encode it in the collection path rather than via, say, metadata?
Because one of the decisions that the science team made was that we don't want to go into the files and anonymize them. Our objective, so first of all, it's very hard to anonymize the raw data because it's, again, on the other side of an FFT from an image. So it's almost impossible. With the DICOMs, some people go in and deface the MRI images and such, but our science people don't want the files modified from how they come off the machine. So, and thus putting the experiment in the name field means that they never have personal identifying metadata in them. I mean, you can debate whether an MRI image is personally identifying. I mean, people have done facial recognition off MRI images. So, but I try to, like, we have a data privacy representative at the Institute, and then Max Planck, the group of institutes, has a data privacy thing. And, a lot of GDPR hasn't been litigated yet, so you can get different. I mean, you have to draw a box around the system at some point, and that seemed reasonable to us, and people like it.
So the thing that I was thinking with that is that if we have an incrementing number that's just going up, you can end up with a lot of collections in that sub-collection. And it's something we face in one of our zones, and it's a design choice that possibly we wouldn't make again. So when you get to the hundreds of thousands of collections kind of thing, it's not a good look. So something to keep an eye out for.
That's why the study ID is at this point in the path. And where I have an iRODS structure holding just the experiment IDs, and that's done with the, collection for the first letter, collection for the second letter, collection for the actual metadata. So I've tried to avoid that. These things are, these experiment IDs are actually encoded to be error correcting, so that part of it's the number and part of it's a hash on the number, and then it's converted into alphanumeric, so that if someone mistypes one of the letters, it's going to, it should be refused. I think it's a 1 in a million chance to, make that mistake and get it right. So I think we're okay with that. The thing that's getting us right now is that I'm uploading DICOMs individually. And so, when you talk to the scientists, they give you one number for like, what's the maximum number of files and what's the max. And then it's of course an order of magnitude greater when you turn the thing on. And so that's where we're running into the very high file counts. And that we're going to deal with more heuristics that pack the DICOMs into a tar file before committing it to iRODS, which means this part about how you're not sure when you're done with the scan, that just, it becomes more churn, because if you get a late one, you have to go get the tar file and, you know, add the thing in. Yeah. So I think we're going to avoid massive numbers of objects in a single collection. I think we got that.
All right. Oh, we have one more question. And then we have to head outside to take a picture.
Hi, yeah, I noticed in I think it's a previous slide that you run iRODS production in a Docker container. Have you always run it like this? And do you notice any performance penalties with running it like this?
Well, no, as I mentioned, the first deployment was running everything in a VM. What I have never done ever, my iRODS experience, is installed on bare metal. The VM was a mere VirtualBox VM, you know, not commercial software, but we had essentially, it seemed like we were limited to gigabit Ethernet bandwidth in and out of it. And basically playing with it didn't yield higher bandwidth. And I've been kind of thinking about Docker because a lot of the iRODS ecosystem is made available, like I say, with Docker. And the performance troubles we were seeing with iRODS went away immediately with I mean, I tested it before we did this, but the performance, the IO performance went away immediately with the Docker container. Maybe we'll run into something else. If anyone has really bad experiences with iRODS and Docker, do hunt me up and tell me what's ahead of me. But so far, this has been running really well. When you update one of these guys, you run the Ansible, it pokes at the production server, it starts a new container and keeps running.
All right, thanks.
All right. Thanks, Blake.
Thank you.